Well, good afternoon and welcome to MedTech Crossroads on this Friday, July 24th. And I got a lot of guests today. I'm so excited to have everybody here. First up, we're going to have John Cunningham and Angie Purvis from RapidBio. And then we're joined by Sandra Cochrane from WMED Innovation Center. And then finally, Bill Worzel, who's a longtime guy in the startup community doing another startup coming out of MSU. And so I just wanted to mention one thing. I'm remote today, and as you know, we famously are not looking for sponsors or anything like that, but today I have to acknowledge that the remote broadcasting facilities that we're using to bring this show to you are donated generously by Rathburn Contracting, so we're thankful for the space to hold this despite the distance. And what we're going to do is we're going to kill this screen share, and we're going to welcome our first guests, John Cunningham, who is the CEO of RapidBio, and Angie Purvis, their lab director. John and Angie, welcome to MedTech Crossroads. Good to have you.
Yeah, thank you very much. Yeah, thanks.
Well, we're pleased to have you here. And I think what we're going to do is have you start off with a little presentation about RapidBio. What you guys are doing is rapid COVID diagnostics in the local area in Southeast Michigan. I read somewhere that you guys are shooting for 6,000 samples a day is where you want to get to.
That's the goal. Maybe it's a stretch goal, but that is the goal, yeah.
That's pretty cool. Well, since it's very germane to something that we've told our audience, we're always going to talk about when there's news about it. And since you guys are right at a neat inflection point for that, I would love it if you'd bring up your presentation, talk to us about the state of COVID testing and about what RapidBio is doing here in Michigan to help.
Great, sure. Okay, how do we play this? Yeah. Well, Gene, thanks for having us. So yeah, so Angie and I are working with a new startup company called RapidBio, and we're focused exclusively on COVID testing for the foreseeable future until there's no longer a need for the testing. And we started the company basically in April. So it's been really a roller coaster ride from then till now. So just wanted to share a little bit about our story and talk a little bit about what we have planned for the future. So just as a refresher, there's two main types of COVID testing that are going on. The solid lines here represent the molecular diagnostic, the PCR testing for the actual live virus. And there's multiple different sample types. This is an illustration of sort of how, what's detectable over the course of the disease progression. So we're focused exclusively right now on the molecular diagnostic and looking at, nasopharyngeal swabs, and then trying to move into other sample collection types. We have partners that do antibody testing. And that's certainly going to be a very interesting place to be as well, very important, but we're focused on the molecular diagnostics right now. And really the pain point here is just that, yeah, I think is becoming, obviously it's in the press. You can be asymptomatic, have no symptoms, and be contagious for up to 14 days. So this is the sort of, the silent spreader phenomena that we're seeing. So this sort of graph sort of illustrates the importance of having not just diagnostic testing, but also screening of asymptomatic people. And that's really sort of that's really our goal. Just a quick, maybe just a couple of slides to sort of show folks where we're at. So we started in April, formed the company April 9th, applied for the CLIA certificate April 10th. We had the CLIA certificate in 10 business days. It took us almost 2 months to get a lab space. But so we're situated at the Michigan Life Science Innovation Center. We moved in first week in June, and just, it's a great facility. There's, many great facilities in Southeast Michigan, but this is where we happen to end up. It's a full biosafety level 2 facility. This is the core team that we started with. This was a couple weeks ago. We've doubled in size, and we'll probably double every month for the next, for many months. We're actually housed in an old vivarium. Milsic facility was originally used to develop asparaginum therapeutics. So we're in this vivarium space. This view has become sort of I've become accustomed to this view. This is my home turf now. But we're using basically a series of these very small cage rooms, which is great for molecular diagnostics. So these rooms are all negative pressure, 12 air changes per hour, etc. So just walking you through the process, we have a partner that just, I picked up 600 COVID samples from a partner the other day. So we have a -80 freezer filled with COVID samples, and just walking you through the process. This is a biosafety level 2 hood. We have 2 of these now up. So this is where the live virus is put inside this hood. The cap is opened and the sample is lysed and basically inactivated. And then we move on into the process from there. This is Angie here modeling our new lab space. We have a total of 5,000 square feet of lab space. This is a 2,500 square foot wet lab that's mostly empty now. We're using it for a manual RNA extraction process. So we have a process where we can manually go take the samples and extract from the nasal swab sample, extract the RNA. It's about a 2-hour process of wash steps and heating and adding magnetic beads primarily. Ultimately, we want to automate that process. Our goal is in a very short order to use these robotic liquid handling machines. We have 3 of these machines basically on site, and we're just getting them up now. I don't know if we have time to show a little video, but actually maybe I will. Do we have time, Gene?
Yeah, go ahead and give it a try. It's a short one. Yeah.
Yeah, sure. Okay, so how do
we love the graphics, the visuals.
So this is a 96 pipette head pulling up 96 pipette tips, dropping down, extracting 100 microliters of fluid, and then moving over and basically sucking it up and then dropping it into a microplate. So this is the robotic liquid handling process. There's a 2-hour dance basically this robot does to simulate what the humans would do. So anyway, so that's just a quick view of what we're working on. And then back to this slide deck here. So from there, we go into a preparation step where we basically, sort of prepare the RNA samples for the PCR. This is an interesting illustration. This box will analyze 1,000 samples, and it costs $18,000 for this box. So
wow.
You don't want to misplace this particular box. This is the Quant Studio 6 PCR that we have up and running. We have 4 additional PCRs in crates ready to set up. But so this PCR will basically process 384 samples in about 2 hours. And we'll basically it's like a bread machine. It basically heats and cools and heats and cools and heats and heats and heats, and then it shines a laser through each well, and it basically gives a readout. And then to wrap it all up, we have a $250,000 lab information management system that's being deployed next week. So that's sort of like a visual tour. Where we're going to high throughput saliva as a sampling method, pooled testing, and screening of asymptomatic people. I've got a couple of visuals, and then maybe we could open it up or have a discussion about the regulatory environment. So fundamentally, right now, the primary sample type is this nasopharyngeal swab. This goes all the way to the back of the nose. People are validating a mid nasal swab, and then there's work being done. There's already groups that have validated saliva as a method. So that's where we want to go as quickly as we can, assuming we can get it to work as well. And then the next piece is really pooling. So this is an illustration of the pooling concept. So instead of running, if it costs $30 in reagents to run one sample, instead of running 100 individual samples, there's already folks that have validated that you can basically pool, take 5 samples or 10 samples or even 20 samples, pool them into one run, and then you can basically report all the negatives out. And then if you have one pool that's positive, then you can go back and look at that pool. So that's the pooling concept that's now, Dr. Fauci's been talking about it, and Beaumont just validated it, just submitted an EUA for this, and other groups have validated this. So this is where we're going, is the pooling.
So this is how you screen through a data set. You chop it in half, you look at one half, and if the thing you're looking for is not in that half, you forget about that half. Is that roughly?
Yeah, essentially, yeah. I mean, I think the typical pool size is 7, but we have a partner that's trying to validate 20. But both to save reagents and cost and time, if since the analytics are so sensitive, they can pick up very, very small fragments of RNA. So in this illustration, you can essentially test 10 at a time, and drop your cost dramatically. And this is the kind of methodology that would make it cost effective to screen your employees once a week. So yeah, so that's sort of pretty much the end of the visuals. Any discussion, Jean, or do you want to add anything in, Angie?
No, it's really great. If there are questions.
Are there any questions? Or
John, help me to understand in terms of the local area and what you guys are going for. Where is most of this testing being done right now?
there is a state. The state's doing a really great job of organizing the local labs. There's 30 or 40 labs in the state of Michigan that are running, and there's a schedule that comes out every week with all the labs and how many tests they run. It's really a combination of, you've got the major hospitals, all have very good testing capacity, but they're primarily focused on their own internal customer base. And then you've got the national labs, the LabQuest and the LabCorps and the Quests that are running. And then there's a whole set of small independent labs that are actually serving a lot of the high volume work that's being done is done by small independent labs. So we're hoping to sort of join that community of small labs. Yeah,
that's a phenomenal resource that you guys are working to bring here. Let's see, we did have a couple of questions live. These are from our friend Francis Glory. And again, I don't know what of this you're comfortable answering or not, but the two questions were, are you using life magnetics for RNA extraction? And the second one is, do you have a major partner supporting your scaling up?
So yes, we are using Life Magnetics, has been a partner since day one, and really sort of helped to lay the foundation for for the lab. We are validating Life Magnetics RNA extraction technology, in addition to the Thermo Fisher kit, and there's several other kits. Omega, Promega, and Zymo are the main kit manufacturers, but we're excited to work with Life. We also have a project working with Acadium as well to validate their technology for this application. Oh, partners. Partners. So we have right now the main partnership is we're partnering with another local lab that's providing us with. We're sort of really. Another local lab that's very well established in the market, but that needs capacity. Yeah.
Very good. John, could you take us back to a graphic that you had? I think it was almost your second slide, yeah, where you had the timing of infection for the disease. Can you walk us through that one in just a little bit more detail? I think this is something that folks are fascinated by. Of course, you know, you'll overhear these chats and people will say, oh, I got tested, so I'm fine, or oh, I got tested and I've already had it, or I think it was the next slide after this.
Oh.
This one here. It seems like there's a boatload of information in this slide. And I think one of the questions that people have just at a lay level is, what do I know and when do I know it? A number of months ago now, we had Dr. Ing Pottinger on the show, and she talked through the number of tests that it sometimes takes to get certainty in terms of false positives, false negatives, positive predictive value, negative predictive value. And when you tell people that, they go, oh, I thought I just took the test and I got a binary answer, and that was, you know, definitively the truth. And then on top of this, you've got this sort of hidden period. Can you walk us through, for those folks who are really sort of even at a lay level wrestling with this and going, okay, I get it, not every test is 100% accurate. On top of that, walk us through these windows again.
Yeah, I think, yeah, well, just on a personal note, so my daughter's summer job at the Argo Livery was shut down because one of her coworkers was at the 4th of July party where 72 kids were invited. So she got the test, and then she emailed me, you know, 9 days later and said, I'm negative. And I said, well, at least you were negative 9 days ago. Right, so yeah, so and again, both of these slides, these are both illustrations and approximations, and they were published at a point in time. So you know, so in the data's you know, constantly evolving, and I'm certainly not an expert at this particular, you know, in this area, but the basic concept here is that you know, people say it takes, you know, 5 to 7 days to see symptoms, you know, from the point of infection. But you know, and I guess the question is, you know, where is the infection seated in the body, and at what point is it basically expressed in the respiratory tract to where you're shedding the virus? So in this graphic, you know, we're saying that you know, that only a small fraction of the infections become critical at all. So 5% or less would actually become, you know, would be you know, severe. Would be critical or severe, and the vast majority of the cases would be in this sort of moderate to symptoms to no symptoms range, where you could say 80, 90, you know, +% of the cases, you know, I think many of us have friends that, you know, were positive, but really had very few symptoms, right? But then the question is, how long how long are you shedding virus before your immune system kicks in? You know, and I think that I don't know if we know what the current you know, literature is on this, but we think that it could be 14 to 21 days that you're shedding virus.
That's very good. That is a very, very interesting graphic, one that I hadn't seen in this particular form. So you guys are ramping up, and you know, again, from a lay perspective, is this something that people are going to be able to get access to in the future? Can we come directly to RapidBio, or are you guys servicing directly through hospitals, or how does this work?
We'd like to have a B2C capability, but it's probably later into the fall. I mean, certainly, you know, we're happy to take one-offs, but in terms of the IT infrastructure to make that happen, I think it's going to be difficult. Our main sort of like market thesis is direct to employer. So direct to organization, direct to employer, large organizations that, you know, that want to have like a once a week screen and get back to work screening capability. You know, our current thinking is to be more of a wholesale provider. Because we don't necessarily at this point have, you know, 50 nurses in vans driving around, you know, or setting up the, you know, the capital to set up collection stations and things like that. So we may, you know, our primary model may be, you know, servicing other labs that are already over capacity, or large organizations that want to do a large sort of batch contract. The call this week with the lab directors, the sentiment was there may not be availability of testing for screening because we're still in the first wave. If the first wave doesn't drop off pretty soon, the first wave is going to hit the fall and flu season, and then we're just going to be in this. The comment this week was we may only be able to have the reagents to test sick people. So there's a question about whether or not the screening capacity is going to be there. Certainly, obviously, if the disease continues to ramp and incident, if the disease continues to outpace our ability to add capacity, it's going to be an issue.
That's very, very interesting. I guess it is good to know, though, you guys are, what's that?
Which increases the importance of pooling.
Which increases the importance of pooling. That's interesting. I think it's also interesting to know that you guys are going B2B in terms of large companies. So large companies who might be listening, that's an open avenue of discussion. It sounds like you're not just going to hospitals, but you could do B2B with commercial companies.
I think the hospitals are pretty well served with their own internal capacity, as long as there's not like a breakdown in reagents. All the major hospitals can do 1,000 or more a day. So I think they're pretty well served.
Okay. We have a question here from Reiner. He says, are there shortages of reagents and swabs in Michigan? I guess currently, you alluded to something in the future, but any knowledge of reagent or swab shortages in Michigan at the time?
Yeah, there are. I mean, there's multiple different vendors. There's multiple different, reagent kits and hardware manufacturers. One of the major manufacturers had a lot failure 2 weeks ago, and so the entire nation was shut down for maybe 5 days. And as a result, some of the large labs were potentially shut down completely based on that. But I think it's lumpy. But I think overall, the kits are shipping, and so there are
Okay.
reagents. And then there's a question about the comment this week on the lab call was, that the federal government is coming in and basically jumping to the top of the line, and we're not exactly sure where the kits are going. But I think there's also I know that there's a focus on the hottest states as well. So I think at a national level, there's an attempt to try to direct capacity, to Florida and Arizona and Texas and places that are really, really in trouble right now.
That makes sense. That's a good point. Yeah, great questions there. Any other questions, please do raise your hand or type them in. I guess one more thing I want to ask you, John and Angie, is in terms of where you are right now and what you have left to do before you reach capacity, how can the community help you? What are you guys looking for? What are your next steps? What sorts of connections are you looking to make?
Well, we're just closing we've raised $1 million. And we have a term sheet for a follow-on round of another $1 million. And there may be space in that. I'm not sure. We are looking for molecular diagnostic technicians, and also more like entry-level laboratory technicians that have experience in clinical labs. So certainly we're looking to add. We could be adding 10, 20, 30 people in the next couple of months, and we're looking for people, experienced people in particular. We are looking for partners for these studies. So partners that would like to work with us on collecting matched nasopharyngeal and saliva samples, and would like to participate in a large-scale screening and pooling studies. And also potentially some additional resources to help us with study design, epidemiologists, and people that can write IRBs. Those are the main asks.
Great. Well, certainly if people are listening and have any links to make, that would be most appreciated. All right. Well, let's see. I'm not seeing any more questions here. Is there anything else that you'd like to tell our audience before we go, John and Angie?
No, I don't think so.
Well, we greatly appreciate your coming on today, sharing this with the community. It's much needed, and there's Grandma with the thumbs up. That's great. I love that. We appreciate what you guys are doing, and appreciate your communicating the current state of things with us. And we would love to have you back as things progress to tell us what you've been finding as you start to really get into these samples.
Yeah.
So appreciate you being here.
All right. Thanks,
John Cunningham, Angie Purvis. Thank you for being with us.
Thanks much. Okay. Bye-bye.
That's awesome. It's fun to see local companies working on these national problems. Interesting point though that John made about the potential for reagent shortages and Reiner's question about reagents and swabs. So I'm sure we'll hear more about that. Next up, I want to welcome Sandra Cochrane, who is the Assistant Dean at the WMED Innovation Center in Kalamazoo, Michigan. That is one of the smart zones. Hey Sandra, how are you?
Hi, I'm good. Thanks for having me today.
It's so good to see you. We've had a number of West Michigan folks, and today I'm actually sort of on the west side of the state, so we're just leaning it over there as best we can.
Well, it's a beautiful day on this side of the state, so welcome to our neck of the woods.
Thank you. It's beautiful. So you guys, under normal circumstances, you're a smart zone. You're an innovation center. You're doing, and I've been over there a number of times. Beautiful facility, beautiful programs. I remember one that I was over there with you guys for. You had the USPTO in for a meet and greet and talk, and that was a wonderfully informative thing. And tell us just a bit more about. We're going to go into where we are today with COVID and shutdowns and things. But tell us just a bit, Sandra, about WMED from a 40,000-foot view.
Sure. So the WMED Innovation Center is about a 69,000 square foot wet lab facility primarily. It looks a lot like MILSIC, which John just talked about, the Michigan Life Science Innovation Center. We're very, very similar to that. So we've got about 76 laboratories inside our building, a whole bunch of offices. We have meeting space and conference rooms and public spaces. So it's a fairly large building, and we do a lot of what they would do over there at the MILSIC. So we have clients that lease space from us. We have 3 separate co-working spaces, which we've established to help the even earlier stage companies. And we do a lot of outreach, so training, education, networking. Well, usually we do a lot of that kind of stuff. We've been a little curtailed lately, but we work hard to support the entrepreneurial ecosystem in Southwest Michigan. And so we are reaching out into kind of a 9-county region around the Kalamazoo area, Cass, Van Buren, St. Joe, those kind of places. But most of the work that we do is here in Kalamazoo. And we love to bring people in from all over. So people don't need to be from Kalamazoo in order to take advantage of some of the training opportunities and space that we have available.
Yeah, that's really cool. And like I said, it's a beautiful space you got there. But here we are, and we're not in that space, and none of us are in these spaces. And how are things going right at the moment in terms of supporting current clients?
Yeah, so the current situation has surprised me a bit. We're actually doing quite well. I was worried when COVID hit and a lot of things were shutting down that my clients would struggle. We'd have a bunch of people maybe going out of business, things like that. And that has not happened at all. In fact, several of my clients have actually expanded during this time because a lot of them are kind of addressing some COVID-related activities. And so they've actually found a need for more laboratory space, more office space. And so we've actually been growing a bit. And I understand from my colleagues around the country that that is not unusual. A lot of the life science specialty programs like mine around the country are also experiencing that kind of surge in growth because there's so much attention on that kind of work right now. Whereas maybe the more mixed use type of incubator programs, they are losing some people as the COVID-19 situation is affecting small business, solo entrepreneurs who are doing like business to consumer and consumer product types of work. So we've been doing very well. So our clients continue to operate, their laboratories are running. In terms of the support, we've not been doing, clearly, no client lunches, no monthly hangouts like we normally do. So I've been doing a lot more one-on-one, a lot of emails with clients, a lot of telephone calls, a lot of video conferencing. We use the Teams platform at my work, and so we do a lot of the video conferencing. It works pretty well.
Yeah, I think we've seen that similar sort of thing where it's like, oh, there's a lot happening. There's more happening than you thought there might have been when this all came down. But as you and I were talking in preparation for this, I think one of the things that we both kind of went, yeah, the next hill over the horizon here is this. I don't feel like I'm meeting as many new people. I would have gone to conferences, I would have done this, I would have done that. I mean, it's great to be connecting here. We've met a lot of new people through these kinds of forums, which is awesome. But what do you make of that? What do you, as you look to the future, what are you trying? What are you doing? And what does it look like going forward in terms of events and things like that for you?
Yeah, we're putting as many things virtually as we can. So we still host 1 Million Cups, which is a kind of a community building pitch event once a month. We're still hosting those kinds of things. Whatever we've been able to put virtual, we do work with BBC for SBIR training programs. We usually host them live. Those have all gone virtual. In fact, I've contacted them and said they're going to be virtual through the end of this year. We won't be doing any live events. So, things are going to slow down a bit because we won't be doing things live. We'll be doing things virtually. And I'm not sure how many new people necessarily come to virtual events because it's difficult to build a community virtually like that. There's something to be said for being live with someone, standing next to them, having a drink with them, sharing a lunch table and eating lunch at a conference. You're going to talk to all of these people who are sitting around you that maybe you wouldn't have talked to. And so it makes individual conversations like we're doing now a little difficult. I've noticed like on our call, our participants, we've got 26 participants and I'm probably not going to get to talk to any of them. So even though most of them are new to me, I'm not going to be able to really connect. And that makes it really hard. So in the future going forward, we're hoping that either everything, we hit that plateau and everything tails off and COVID becomes a little less, devastating, or we get the vaccine and we're able to start doing some live events. So I've actually taken all of my events that I was going to do this year between, March and the end of the year. And I'm trying to see what all I can shove into next year. It's going to be really busy next year. So everybody, we're going to have so much stuff going on.
Well, since you're shoving those into next year, tell us a little bit about what's going to be happening next year.
Oh, wow. Okay. So like our big event that we were going to hold in November, which we've pushed off till June, is called the Boundless Women Confab. That's actually being funded with some money from the Small Business Administration. Was to support women entrepreneurs, especially in the tech sector. So we are having a full day event on June 2nd. We've moved it into next year, and it's going to have a professional track, a professional development track, a personal development track. We have speakers coming in from outside of the state of Michigan, but a lot of our sessions are being given by people who are in the state of Michigan. It's going to be just a great day to celebrate women and entrepreneurship. We anticipate that a lot of the attendees will be women, but men are certainly welcome. And we would hope that they would come to kind of learn about what's happening with women and entrepreneurship, especially in the tech sectors around the state of Michigan. So that's one thing that we've got going on. I'm working on a strategic planning type of a workshop, a 3-part series workshop that we were going to initially offer in December of this year, but I'm going to push that off now into next year for strategic planning, people who are interested in doing that. We've got our SBIR training program. We're going to continue that. We work in collaboration with BBC, and we'll continue to offer those programs, hopefully live again, where we do more lunch and learns. Yeah, so those are some of the things that we've got going on. There'll be lots more things, but those are the ones off the top of my head right now.
That's great. Actually, you said you weren't going to get to interact, but Jeff Reinfeldt just asked, do you have a URL for the events? Is there a place we can point people to for the events?
Right now, we currently have it on our calendar, which is at med.wmich.edu, and we are under the research tab. And so under the research tab, you'll find the Innovation Center and all the things that we do. And we have our calendar out there. We don't have a separate URL just for the event, at least not yet. Maybe we will in the next.
That's great. med.wmich.edu under the research tab. Well, that's great, Sandra. I really appreciate your coming on, telling us a little bit more about WMED and what you've been up to. And I think this question of how are we going to keep meeting people? How do we, at very least, it means we need to keep reaching out and meeting those new folks, that next year's worth of people that we need to be interacting with. And maybe also it sparks in somebody the idea of there's got to be a better answer. We've got these constraints, but there's got to be a better answer. How can we keep that part of it going? But I'm glad to hear that things are trucking along well, and you guys are still offering what you're offering. And like I said, great facility, great team over there. So thanks for being with us today.
Great, you're welcome. And yeah, I hope people, if anyone has any questions about us specifically, you know, reach out to me. You can find me, you can Google me, whatever, and happy to answer any questions that anyone might have. So thanks.
Yeah, and we'll actually pause here really quick and just say, does anyone have a question? Because we can take it live right now if anyone's got a question for Sandra. Actually, here's Ken Spencer. Ken, you're live.
Can you hear me now?
Yeah, we can hear you now. You're great.
Okay, thanks. So just wondering if you've been in contact with Jen Baird for your series on women in entrepreneurship.
Yes, I know Jen quite well. She's actually, we did something else we had to cancel that we might pick up next year. We had a whole series called the Medical Entrepreneur Speakers Series. And Jen was one of our inaugural speakers for that particular event. And yes, we will be reaching out to her when we do our Boundless Women Confab.
Great, thanks.
Yeah.
Love it. Thank you, Ken. All right. Well, not seeing more questions. Once again, Sandra, thank you so much for being with us. I hope folks will reach out, and do wish you the best, and hope to have you back sometime next year to hear about these events again.
You bet. Thank you. Good to talk to you.
Thanks again. Bye-bye.
Bye.
Yeah, it's really fun looking at these smart zones around the state, and I know that the state has put a lot into them, and it's really fun to see the niches that each is finding and yet working together. It's turning into quite a nice network. My next guest, Bill Worzel, is with us. I want to make sure Bill can get his video and audio going here. Bill is a computer scientist by training. There he is. But he spent a great deal of time over the last 20 years using machine learning to analyze biological and medical data. And he's developed quite a few different diagnostics and prognostics for the market. And Bill's currently working with a startup company coming out of Michigan State University, which is using machine learning to make a better hearing assist device. And Bill, is your audio working?
Yeah, it is. Can you hear me?
It's great to see you. Yeah, you're just fine. Welcome to MedTech Crossroads.
Thank you. It's a pleasure.
Yeah, well, you and I have bumped into each other and worked with each other on many different occasions, and I thought it'd be fun to have you come on, because when I think of Bill, I think of the word prognostics, and I think a lot of people know what diagnostics are, and but give us a working definition of prognostics as we start this conversation.
Sure. So diagnostic says you're looking for something, do you have this disease? A prognostic is the sense of trying to predict what will happen in the future with, say, a disease. And so if we were talking about COVID, and I'm not doing any work with COVID, so full disclosure, diagnostics would say, well, you have COVID. And prognostic might say, you have COVID, and you have a very indolent form of COVID. So we can take things that are more, easily in that situation, not that you take it lightly. So that's the difference between diagnostics and prognostics. And really, diagnostics is a big market, but it's usually a very simple process. Prognostics are a little bit trickier because unless there's a single marker that says, yes, this is, this is how it's going to, this is going to drive what's going on. It's often a multifactorial thing that you have to hold together different pieces of data to do it. So that's kind of a quick quick and dirty thing about it. Prognostics are usually much more interesting because they help doctors make decisions often. So if I had something that said you have an indolent form of cancer, a doctor might be very hesitant to pull out all stops. But if it says it's a very aggressive cancer, the doctor would say, we got to get on this and throw the kitchen sink on. Now I'm all the doctors on there are now shrugging, making faces at me, but I'm just saying that as an example.
Well, this links back to even what John was talking about earlier in the show when he showed us a graph and he said, okay, here's a stage you can be at, or here's one, and you might have different symptoms up here versus down here, and only this percentage actually make it down to this really critical state. And what you're sort of, I think, telling us is, prognostics is how do we get from this level that we're at, and again, we're not specifically talking about COVID here, but we can sort of relate the learnings to COVID a little bit. If it was diagnostics, we'd be saying, do you have it? And if it was prognostics, we'd be saying, is there anything we can say about which of those categories you're in right now? Which I think is obviously where we want to get to.
Yeah, and there's also the case of whether you can say that this particular treatment will work for it. So that's another important part of it, that you're predicting whether that particular thing is going to help with that disease. Sorry about that. I lost the word.
Yeah, no, it makes sense. Well, so prognostics is largely based on modeling, building models of things. And I know when you and I chatted before, there's a couple of interesting things when it comes to modeling. I know in my life, when I've submitted models to FDA, their first question is this model validated? And of course, we've recently seen a lot of models in the public eye about what's going to happen with COVID cases based on this and based on that. But tell us a little bit about your view on modeling and what the conditions under which modeling is useful and under which it's not useful, and what the limitations and strengths of building models for these sorts of things that you've built models for.
Yeah, well, if I go back through my entire litany of things I've done, which I won't, I've done things as far as modeling rocket fuel for JPL. Okay, and when I moved into and I've done other things in other areas, but when I moved into biotech, I was mostly doing a couple of things, modeling a disease progression, which we sort of I alluded to before, modeling what the likelihood of a particular treatment will do for a particular disease or a particular individual's disease. And, from there you can go as far as, what your data takes you. And so, as you said, I'm a computer scientist by training, and so I got into this, backed into it, looking for a new job when my last one was bought out by Google, and which I got a very small piece of. So I got into it because I was naively walking around thinking, hey, this biotech world has all this data. And one of the things you have to understand about data is you may have a lot of data, but there's not a lot of differentiation in a pile of data, it's very hard to pick a signal out. So, we ended up working towards places where there was clear data and clear differentiation of the outcomes. And so we were working a lot with cancer, so we could say, for instance, this is a cancer which is fairly indolent, this one is more aggressive, or we could be, in certain cases, we worked with saying this drug is likely to work because of the nature of what we can find from analyzing it so this drug will stop the disease at a key point. And this is, that's what everybody wants to do, but we were the ones that were, in some cases, figuring out exactly where that point was, what the key point was that you wanted to attack from a disease. So we could repurpose drugs, and we also did some work in developing new drugs, at least targets for drugs.
One of the perspectives that I know you and I chatted about was the use of modeling as a generator of questions, which I thought was just a fascinating way to think about it, because I think a lot of people go, I'm going to build a model and it's gonna predict the future. And one of the things you told me was, well, it's actually it's one of the most powerful ways to generate new questions. Tell us a little bit more about that.
Yeah. So it's you can often start out if you're really going to work on this sort of thing, you can start out looking for one thing, and then something else can come up out of it. And this is, since I'm in bioinformatics, this is something I always look at, which is to say that I want to look for the thing where we do an analysis and it says, that's really interesting. Why is that happening? Because in honesty, I've done some reading on this, and I'm not saying this is what's going to happen to me. An awful lot of Nobel Prizes are awarded for people who, something happens in an experiment, and most people just sort of threw it out and said, well, it didn't work, because they were so focused on what they were trying to prove. But a lot of Nobel Prizes have actually been awarded when people said, I wonder why that happens. And they dug into it and they found something, very extraordinary. Now, that doesn't always happen, but it's something that you look for, and if you work the way I do, because the data is always telling you something. It may be telling you you're asking the wrong question, and that's tremendously important.
Yes.
Okay. But a lot of people will say, oh, it didn't work. And so they lost, a really important opportunity. So that's
interesting how model building really finds its way into all sorts of disciplines. Here you're working in these data-heavy situations where the data is there, and you can build these complex models and then find out, am I asking the right questions? Am I getting answers? Can I correlate those answers with things? It does strike me that even in other disciplines, there is this sort of model building thing, although we don't think of it that way. I mean, even just the process of planning a project is sort of a model building exercise. Then you go to execute the project and things don't work out the way that you thought they did. And you have to say, well, where was my model wrong? And sometimes if you're open to that, you'll learn something powerful about the actual inner workings of your team, your project, your product, et cetera. So it's a very, very interesting insight. I liked the way that you brought that up. Well, Bill, so you have been in innovations and in companies for years and years, and you are now working with a startup coming out of Michigan State University, Nimble Acoustics. Right. Would you tell us a little bit more about that effort?
Sure. So this is a company that is just getting started, and they're working on hearing aids, is what you call them. We call them hearing assistants for a variety of reasons. And so the thing about hearing assistants is that you have to tune it. I mean, if you get a hearing aid, if you have to have a hearing aid, anybody you've seen them. They're always fiddling with the hearing aid. They move into another room that's had different acoustics, and so they start poking around at it and trying to turn it and so forth. And it's incredibly annoying, and it sometimes leaves people sort of saying, forget about it. you know, and they just try to guess what people say, you know, and we now have an aging population, and so this is becoming more and more of a problem. So a group of people up at MSU have started working on using machine learning, in particular deep learning, to automatically tune the hearing assistant. And they've been getting some very interesting stuff, and they brought. I'm working on that now, in terms of, you know, how to make it into a product. They have a great technology, but as all of us who've gone down this road know, that's the beginning, not the end.
Yeah, very much so. Well, it's great, great to see you applying what you've done to that as well. We have a question here from Jeff Reinfeldt. I'm not sure where the question was directed exactly, but his question was, and maybe we can reflect on it together, Bill, how does the FDA look at prognostic testing versus diagnostic testing? Can you tell us about that? I've got certainly some thoughts on benefit risk and its basic application, but have you experienced that from the FDA's standpoint?
Yeah, so it's sort of a before and after case, in my opinion. I mean, when we first started doing these prognostics and diagnostics, this was back in the early 2000s, right? And at that time, there was sort of a generational change coming, but it wasn't quite there in the FDA. And so a lot of times they were very, very skeptical unless you could sort of say, okay, here is a protein, that's the signal there is, and that's the whole story. And we were coming in and saying, well, we're looking at this protein and this protein and this protein, and how they interact with each other is how the diagnostic prognostic works. And they didn't know what to do with it. And plus the fact we ran into a problem where we were using a totally different kind of learning algorithm that they'd never seen before. And so, you know, I had a case where we found something really good, really strong. We took it to the guy in the field who also, you know, advised the FDA. And he, you know, listened to what I said. I mean, I literally flew out to Seattle to talk to this one guy because he was the gatekeeper. And I talked to this one guy, and he sort of, you know, listened to me, and then he leaned back and he said, okay, where's the statistics in this? And the technology that I use is not statistical. You use statistics to evaluate it, but it's not a statistical method. It's a machine learning method. Now, back then, nobody knew what machine learning was. Now people, you know, everybody's using it, Google's using it, and so forth. But back then, nobody did. And so he basically said, all right, what's the statistics you're using? And I literally said to him, the only statistics we're using is to evaluate how good our system is. And I could just see the light that went out from his eyes. And, you know, he just was not going to help me at all after that, because he believed that everything was statistical. So, you know, sometimes
this was the way you needed to look at it, and that was just it.
That's right. And there really wasn't a way, unless I was sort of saying, oh yes, this is a new kind of statistics, which maybe if I was a better salesman, I would. But at the end of the day, somebody's going to find out this isn't statistics, and I'm going to be in trouble.
No, that's interesting. That's a great question. As I think about it too, I think what you're saying there is very true, that sometimes people who are in a position to evaluate things will have a preconceived notion. And when new technologies come along, and we've seen this in medical devices, when new technologies come along, it doesn't fit neatly into the bins. And of course, in med device, you've literally got bins to put them into. And if it doesn't fit neatly into the bin, it's always a discussion as to how we're going to get it cleared. And one of the overarching principles is still always benefit risk, right? If there's a big benefit that you can demonstrate, but there's some risk or some uncertainty, then there is an argument to be made that this should be cleared. But of course, FDA is a huge stickler for data, and if they can't understand the model from a statistical standpoint, a better, wow, how are we going to shift that thinking to another one? That's very interesting. Let me reach out to the audience here and see if we've got any questions for Bill. I see a hand raised. Ken Spencer.
Yes, Bill. So thanks a lot. And I can vouch for the fact that FDA is still trying to spell machine learning out there. But we have a number of things going on at U of M there. But I was wondering, one of the great challenges for what we call predictive analytics or prognostication is sepsis. Have you done any work with sepsis at all?
No, I haven't. Can you tell me a little bit?
Well, the problem is that first of all, way back when we first started working on this 5 years ago, it was like 7 out of 10 people didn't know what sepsis was, at all. I think it's become more, the awareness has improved out there, but the initial criteria for whether somebody is, potentially septic or not is like, higher heartbeat, increased temperature. I mean, and the joke around Michigan, and it applies to MSU too, but back in the old days when we had football games, was that on any given Sunday, half the student body would be characterized as septic, So the issue with sepsis is that if you can actually predict that somebody is going to go on that septic path, you can make interventions way early. The problem we have now is that it's like, oh my God, now he's septic, and we literally throw every antibiotic known to man, on top of them, and your body, the organ failure rate is high, and the hospitalization rate is well over 30% for anybody that's had sepsis. So the early predictive capability of that is really challenging, and sepsis actually costs hospitals more money than any other disease that's out there. So
Sorry, go ahead. What was the question? Sorry.
So I was just wondering if you had done any work on it or looked at it at all in your past.
No, but I did something that was somewhat similar, I think. I had a Dutch gentleman who approached me because they were looking for a test for Legionella pneumophila, which is a pathogen which, has many varieties, and most of them you can literally swim in them, and we do. When you go out to a freshwater lake anywhere and you swim in that lake, you are bathing in it. But there are a few strains which can cause severe illness and even death. And you may remember a while back where you go to the supermarket and you have all these, you still have the trays, but they changed what they did with them. Trays that catch water every time the sprayers come down on the vegetables. Okay, and that water sitting there became, a great way to grow bacteria. And one of them was Legionella, which is what causes Legionnaire's disease, and people were getting sick for them, and it was hard to find. So yeah, I mean, if you swim in water, you want to be able to do it, or if you put it, or even worse, drinking water, it's the same thing. And so we actually made a diagnostic to say, yes, this is here, and it's the virulent strain of the disease, of the biology, the device. I'm thinking devices all the time now. So it's a very virulent form of something, and you have to take some steps with it.
Yeah, thanks for the question, Ken. Appreciate that. Bill, thank you.
Yeah.
Let me just check here and see if we got any more questions. Seeing none at the moment, Bill, thank you so much for being with us and talking about modeling and prognostics. And we wish you the best with your new startup doing machine learning for hearing aids coming out of Michigan State University.
Yeah, it should be fun.
Thanks again, Bill.
Thanks. Take care.
Bye-bye. Well, that's great. Three or four guests today, and what great conversations. It's fun to see what everyone's doing around the state in MedTech. I'm going to give just a minute for any question and answer, for any check-ins. One of the things that I think folks maybe have not been clear on is if you do have an effort that you're part of, when we started this, you are welcome to check in, take 30 seconds, tell the group assembled what you're working on, especially if we haven't heard from you in a while. We'd love to hear from you and find out. You can just raise your hand in the Zoom meeting. We'd love to hear from you. Also, any questions that we can talk about or answer, we're happy to answer them. I'll give another 30 seconds for that if anyone has anything that you want to pop in and say. And we just got some kudos there. Thank you from Kevin. That's most appreciated. And otherwise, I just want to thank you for being with us on this 18th episode of MedTech Crossroads. It was a great show. We're looking forward to next Friday at 2 p.m. And we'll be back with you from the usual location. Thanks so much and have a great day.